Okay, well, I'd like to thank you for everyone here for the hospitality that they've shown me so far, particularly Maria Path, and the opportunity to come and talk to you about this work that I'm doing. I'm afraid my Spanish is non-existent, so it has to be in English. So this is work that Mark Thomas and I have been involved with running for the last few years. Mark Thomas he visited here a year or so ago, but he's just retired at quite short notice and left me to present this to you. So I thought I would first of all give you a quick introduction to CSIRO. So this is who I work for, and CSIRO is the federal government of Australia's national research agency. So it works in all areas of science, basically, everything from agriculture and food, which is the unit that I'm based in, through to radio telescopes and climatology and everything else in between. It also has quite a strong information technology component, which has been called the area called Data 61, and so we work with them a lot with this digital viticulture work that I'm going to talk about. So I also thought I should introduce the Australian wine industry a little bit in case people are not that familiar with it. So recently the crush has been just under 1.8 million tonnes, and it's risen. So 2018 last year was down a little bit, but basically it's risen a little bit over a few years from about 1.6 million tonnes in the first decade of this century. And a lot of that, I think, has been driven by the Chinese imports. So we have a large proportion is just 5 main varieties here: Shiraz, Chardonnay, Cabernet Sauvignon, Merlot, and more recently Sauvignon Blanc. And so you can see the importance of Shiraz in particular there. In terms of where their vineyards are spread out, we're throughout most of Australia, not the Northern Territory, not surprisingly. But South Australia, where I'm based in Adelaide, has the largest planting of wine grapes. But from what I was told yesterday, this is only 10,000 hectares more than Rioja. So it's a big country, and we can see if we look at the actual GIs, this is all of the GIs, there's about 65 GIs in Australia, and even within these larger ones, the area of planting is small compared with the land area. But we can see that they're spread around the south of Australia, and particularly the southeast. We have one area up in Queensland, despite this being really subtropical, and that's because it's at high altitude in the mountains there. Okay, so the work that I'm talking about today is our digital viticulture initiative, and this came from some work in field phenotyping that Mark Thomas and I set up a few years ago. So Mark Thomas is a grapevine breeder. My background, as you heard, is in plant physiology, and so I always have an interest in measuring in the field changes in crop management that we might implement. So in 2013 we got a small amount of money to set up a project to look at field phenotyping in grapevines. So we put together this vehicle. It was self-propelled but manual steer, and basically set up with a Lidar and GPS and RGB cameras. This is quite a high resolution trawling Lidar to get 3D images of the vines, and you can see it was good enough that we could estimate even pruning weights from the data that we got out of it. Now this was aimed to be very high resolution and used as a research tool both in the breeding and in assessment of crop management work. So that was a small project, and that ended, and the industry was interested enough to give us a larger project but with a more commercial focus. So what we wanted to do was to work with people from around CSIRO. So as I mentioned, CSIRO has a very broad group of researchers. So we're working with people in image analysis, in robotics, and even in the mining areas. And what we wanted to do was to really try and use the expertise that we had within CSIRO, particularly in hardware and in data analysis. And these were the main areas that the industry told us they were interested in. And I imagine they're similar here in Spain as well. So the number one was yield estimation, and this is throughout the industry, both for the growers in viticulture, but also for the logistics of the winery and even the marketing. So early yield assessment means the marketers can decide which wines to discount and try and sell on cheap and so on. So secondly, fruit composition. So if we can measure in the field more broadly the quality parameters of the fruit and the maturation, that obviously helps with planning of harvest and the winery intake. Assessment of fruit condition in the field, so this is damaged fruit or diseased fruit. In some areas also they're very interested in canopy size, so a lot of the Australian industry does little or no canopy management. Some areas do more. They were interested in looking at the canopy structure, but even in the areas where there's very little canopy management occurring at present, they were interested in the possibility of having this in a more automated way so that they could actually apply some canopy management on the high production areas in a cost-effective way, which they can't really do at present. Obviously, canopy composition is another component, so if we can optimize the nutrition of the vine, that's useful. And early disease detection in the canopy as well. So in Australia we don't do a huge number of sprays, like averages usually 8 or under, compared with some more humid areas of the world. But still, if we can reduce sprays, it'd be useful. So the first thing I'll talk about briefly then is the yield estimation. And we wanted to do this from early on. So having an estimate of yield at the beginning of the season will help obviously with planning through that season. So in our case, in the Australian industry, most of the industry is not doing crop thinning. We have no limitations on yield, and just removing fruit for no reason is not something that industry is generally doing or wanting to do. So consequently, if we can estimate the number of inflorescences at the beginning of the season, we basically can estimate the number of bunches we've got at the end of the season. And when we look at historical data, about 60% of our season-to-season yield variation is due to the bunch number, with the other 40% being split between fruit set and berry size. So if we can estimate the inflorescence number post-bud burst, we can then use historical data to make an estimate of yield for a vineyard block. And although it will still have some variation to the end of the season, it will tell the grower or the wine company whether they're expecting a high or a low year. So we're actually trying to do this in a very simple way in terms of the machinery. So just using a GoPro camera, same as any people might have at home if they're going skateboarding or something else. And so we just set this up and image the side of the row. So we drive down the row and image the row. So we then take that video data from the GoPro camera, we extract the frames. We've developed a detector for inflorescences using a machine learning process, and that then labels those frames. Then we can put that back together, but the problem is we've got bunches labelled in every frame, and these overlap. The frames obviously overlap to a large extent. So also, as the camera moves down, inflorescence may appear and disappear behind leaves, and so we actually have to track through the video frames. We need to track each inflorescence to ensure that we don't double count it. So we then have a tracking algorithm which then tries to track each inflorescence through the video, and that way we can then count them. So, as with most machine learning work, this requires a lot of hand labeling of images. So we have several hundred images like this. This is one that's hand labeled. If you can see the black boxes, these are hand drawn around each inflorescence in that image. And then this is a output from the detector that's been run on the same image, and we can see that occasionally it's getting it slightly wrong, but on the whole it does a pretty good job of finding those inflorescences. And obviously every season we're putting more data into this machine learning algorithm, so it should be getting better and better over time. Whatever is happening in the background is particularly important, and so getting a broad set of images with different backgrounds is what will drive us forwards. So here's an example video. So this video, this is actually Tanat, and this was run on this video without any images from this vineyard ever having been used for the detector. So the detector itself has never seen any images from this vineyard. And so as it moves along, we can see it's picking up and counting. So this is running at half speed. So the actual vehicle was running at double the speed of this through the vineyard. And we can see it picking up and detecting the inflorescences and counting them one by one. So you see in the background here, it's picked up a sprinkler head and counted that. So again, it hasn't seen this vineyard before, so it's not seen that background, so it's not perfect. But it does a pretty good job of counting these inflorescences. So what we find when we compare with hand counts in the field is that it does depend a lot on that background. So we get from 2% accuracy to within 2% to within 20%, depending on whether that video has been, or whether that data's been seen by the algorithm and what the background is. So when there's a clear sky, it tends to work better. So in terms of yield estimation later in the season, again, using RGB videos, basically the same kind of process, using machine learning to develop detectors for basically all time points from, I guess, pea size onwards. So as soon as we can see the berries, and again, so we can do bunch counts, but we also then have the potential to look at the sizes of those bunches. Now, in the Australian industry, the large proportion of it is using Australian Sprawl vines. So this is basically a bilateral cordon or even a double bilateral cordon, and then with the canes just allowed to sprawl out. And we use that because we have very high sunlight in Australia, and it helps to protect the bunches from too much sun exposure and overheating. The downside for us is that it prevents us from easily seeing the bunches. Like, it's even worse than a heavy VSP canopy with no leaf thinning. So we're going to have to work with detecting bunches that are only partly seen and knowing where they are. So the first part of that, again, is bunch detection. So these are just around pre-veraison. So I think a few, maybe veraison and a few berries. And we can see it's sort of finding little bits of views in this canopy and finding the bunches that are more exposed. So it's clearly a more difficult piece of work for the learning process than the inflorescence counting was. And consequently, it's not quite as successful. So what we're doing now is to, rather than just simply labeling the bunches in the images and then feeding that into the machine learning, we're also starting to do some classifications. So we can classify when we do that, whether it's a whole exposed bunch, whether it's just a small few berries, or whether it's a partially occluded bunch. And so I don't know how well you can see here, but we can see that this hand labeling, doesn't match quite as well the red boxes of the detector algorithm. So the other thing we have here is we've got a much more 3D space than we had with the inflorescence counting, where we can only see some of the bunches from one side, some from the other, some are quite difficult to see. So there's a couple of options we've got to work with that, which we're starting to do. So one of those is to actually use 2 cameras. In fact, we use the 3 cameras in the setup you saw anyway to get different views. But we can use 2 cameras and set them up as stereo imaging, and that way we get depth information. And so we know how far the bunch is, so we can adjust its apparent size for that distance. And also we know that 2 bunches are in the same physical space from that 3D information. We know they're probably the same one. We can mosaic the video frames into a single row, so we get one very large single image. And we've done that with the inflorescence counting as well. The problem we have with that is it's very difficult for the machine learning, because we end up with an enormous image that we have to chop up into very small parts to fit on the CPUs in the high performance computing cluster. And then finally, we can also use the multiple images from multiple GoPros with photogrammetry, which then gives us 3D point cloud with color information. And again, we track exactly where each point is then to within a few millimeters. So those are the things we're looking at now as the next step. The other technology we're trialing is the use of a foliage penetrating radar. And so this is the work we're doing with guys up in the mining, who use it in mining work. And I think this one is quite exciting, but it's also perhaps the one we're least likely to get to work. So we've done some work with computational electromagnetic simulation, where we have 2 leaves, 5 berries, and a bit of cane there with an estimate, so that we can then look at the frequencies that are most likely to be useful for the detection. Secondly, we've set up a test rig up in the shed there, and we can hang bunches of berries of various sizes, and we can put real plants in front of them and real grapevine leaves and so on. And because we know exactly what's there, we can work out what we're seeing. Because obviously the goal of this is to get to in-field quantification of the fruit volume. So we played around with various different radars, a fairly basic 2D scanning radar, which doesn't have the resolution we need, high frequency radar, which tends to get blocked too quick, and a lower frequency radar, which does seem to get better into the vineyard. So this is an example of some scans from the in-house test facility. So this is a single scan here. It's kind of rotated 90 degrees. So the sensor is situated up here, and this is the distance from the sensor as we go down. So what we've got set up here is just a leaf in front of a few berries, and the sensor that we have at the moment can pick up around about 4 or 5 berries from 450 millimeters away. We need more berries the further away it is. So we've got some leaf here and some berries, and we can see we're getting a signal return from both of those. And in the field, so this is a bit of visualization software we developed to work with this. So we tend to put RGB cameras or GoPros onto almost anything we do, just because it's cheap, and we can then go and look at where we actually were if we need to. So this is in the field with a loose VSP canopy, although it might not look it from the images. Again, so this is the distance from the sensor here. This is sensor noise at the beginning. And what we're seeing here is sort of the middle of the images here. So we can see foliage. This signal's from foliage. This signal is from fruit. If we remove the foliage, we can see that foliage signal disappears. So from there to there. But we've still got that fruit signal behind it. And then if we remove the fruit, we can see that that signal's disappeared completely. So we can definitely use this technology to detect what's within the canopy. The big question we have is whether we can get it to the point that we can actually use this as a quantifiable estimate of yield, and so that's obviously where we're trying to go with this at the moment. The nice thing about this is it doesn't use any optics, so it can be used easily in a dusty vineyard environment. Okay, so if we move on to fruit composition, along with other groups as well, and I know there's similar work being done here too. We've been using lab-based hyperspectral imaging first of all to look at fruit composition, and then we're trying to move that out into the field. So I won't go through our whole workflow, but here's basically we have a 3D printed tray with individual berries on it, a reference target. And this just goes through on a linear stage underneath the line scanning hyperspectral camera. We then automatically identify the white target in that. We adjust for the white and the dark references. We detect where the grapes are versus the tray. We then link them to a position because we analyze each of these berries with an enophoss later on. We remove the pedicel. So we've done quite a lot of work on tissue classification, which again we can use out in the field. So separating out fruit from leaves, from tendrils, from canes, from shoot tips, from inflorescences and so on. So we have a large data set which we can use for that. So we can use that to remove the pedicels from those berries. We also take out the specular reflectance. So if we leave the so you can see the shiny dots in the middle here is basically where the light is reflecting back strongly and not giving us data. So we take that out as well, leaving the individual spectra in each of those berries. So we've got an even larger data set now. This is from a data set of 2,400 berries, all of them individually measured in the enophoss after being scanned. And you can see the relationship we get there in terms of sugar, and we have both white and red berries in there. We have pre and post veraison and so on. These are some berries taken from sets of trays. As we go down, they're different time points, and they're just coloured based on the sugar calibrated prediction for each pixel within that image, so we can see the sugar increasing as we go through time. The enophoss spits out quite a lot of data, which we can calibrate against all of those. So we can see we do pretty well against things like pH, total acid, not quite so good against tartaric, and not quite so good against yan, but some of the other acids and sugars and pH in particular work pretty well. This is also a comparison. So this is a training done just on Sauvignon Blanc. So there's just over 1,000 berries included in that. And this is a training done on multiple genotypes, both red and white. And we've got 3,500 berries in this one. So the numbers are the R squared of the relationship. So, 0.96, 0.99 obviously is pretty good. And then in brackets, it's the root mean squared error versus the original data point. So we can see, although it's slightly better when we do a train on a specific cultivar, actually it's only a small difference, and we do pretty well with a large data set and many berries in there. So from that, the next step is to go to whole bunches. So this again is still lab scanning, but whole bunches, and we can see, is these bunches different time points in the season, and we can see this is again sugar prediction. We can see that increasing through there. So we're only just looking at how to take this in the field, and I know you guys have been working in the field a fair bit already. So the first thing was actually to get the camera out of the lab and into the field. And one of the problems that we all have with this is illumination. So our setup that we've tried first is to use, this is a Xenon flash. So it's actually used in studio photography. It uses quite a bit of power, but a lot less power than a big halogen lamp that we use in the lab. And so the setup here is that the flash is triggered at 2 hertz, so 2 times per second, and we synchronize that with the hyperspectral camera with that. So the hyperspectral camera will take many more lines per second, but we synchronize it to take 2 per second with the flash, and we also use an RGB camera in there so that we can see what we're looking at. And so here you can see the flash going off. So effectively then we get a video of individual frames out in the field. So this is a field test, as I'm sure you can tell from looking at that image. So this is basically just run down a panel of vines. So the reason it looks like that is because we've not done any correction for movement here. So we haven't corrected for vertical movement in particular. You see there's a grey shape here that looks like a squash bunch of grapes maybe. But because we've got that classification algorithm that I talked about earlier on, we can take every one of the pixels in that, whether they're aligned horizontally or not, and classify that into a tissue type. So we can classify which pixels are bunches, and then we can run a nearest neighbor type algorithm and find where we've got adjacent pixels that are identified as fruit, and mark them as a single bunch potentially. So you can see this looks like a kind of squished up bunch of berries now. Running the lab-based algorithm on that field data, we get a reasonable range of predicted sugars in that fruit. In fact, this is slightly less than we measured in the field because we haven't optimized the algorithm for the flash yet, although we've been testing that in the lab as well. So the next steps on that that we're probably going to do is actually go to multiple spectrometers so there's two options basically we can go. We can identify wave bands and develop a multispectral sensor. And I haven't got it in here, but for instance, we can go from 150 down to 14 wave bands and still get a pretty good estimate of most of our berry composition. But we'd have to build our own multispectral sensor for that. The other thing we can do is to take many single spectra and use our classification algorithm to identify what they are. So we'll be trying these next season. And again, I know you've done some similar work here in the past. So in terms of the canopy, we're looking at canopy size and structure in a few different ways. So we've done some drone work. We're mainly sticking with proximal in-field measurements, but done some drone work and then used photogrammetry to give us a 3D data set based on the drone 2D images, and that enables us to separate the canopy from the ground, for instance. We've done the same thing with the videos using the cameras on the GoPro cameras. We've also used GoPro cameras and then kept the images as 2D and done gap fraction analysis, which I'll talk about in a moment. And then the final thing we've done is played around with different Lidar sensors. So Lidar is basically a 3D laser scanning. So we're using a spinning Lidar here. So the Lidar gives a single line scan, but by spinning it round, we then get laser returns from all different directions in the vineyard, from different parts of the canopy, and that will go into the canopy and potentially through the canopy, which we can use. We've also mounted it on a tractor, so you can see the same thing on a tractor as well as on our vehicle, scanning up and down. So when we take that data set, we produce a point cloud. This is basically got a mesh overlaid it to make it easier to look at. So here's one vineyard block that's scanned, and we can see the sheds and things at this end, the adjacent blocks here. This is a different one here where we've just scanned a few rows. And then when we zoom in, we can see some of the canopy structure there. This is point cloud data viewed from the top. So this looks like a point cloud. So when I talk about the point cloud, we get each laser beam that's gone out, and that's going at 250 beams per second as it's out there. We get a 3D position of its return in 3D space. And what we're using to put the data together is what's called a SLAM algorithm, simultaneous localization and mapping. And this is kind of almost a self-learning system, so we don't have to use GPS in this because GPS is never going to be accurate enough. And so that's an algorithm that basically learns as it goes through by having multiple hits into the same space and forms this 3D point cloud. This looks like a 3D point cloud, but this is not actually it. What this is we've taken voxelized our data. So by a voxel, it's a 3D pixel. So in this case, all of that LiDAR data, we take then a 12 centimeter cube, and within that, we estimate the density of the grapevine that's in that 12 centimeter cube, and that's plotted out here. So with higher and lower density color coded. Now that in itself is not necessarily what's useful to the grower, but because we've got all of this data in 3D space, even into the canopy, we can look at it in many different ways. So here is a side view of each row, just laid above the top of each other. Maybe we can just see some dots, which is the trunks under there. Here we've amalgamated the data down an entire row. So this is giving us basically the average density and size of the canopy in 3 different vineyards. Here we've got a top-down averaged view. So we can look at this in many different ways and then map that out however we want. So similarly with the drone work, we can use photogrammetry, or structure from motion, which is where, because we have many videos frames that all have a very high degree of overlaid over the top of each other, but from very slightly different angles, we can use a computer algorithm. And in this case, we're using the open source Open Drone Map, but there's commercial systems such as Pix4D available. And you can see that then when we get that point cloud data from the photogrammetry, we can look at where the heights of our pixels are, so and split them out between what's in the canopy and the ground. That can then give us a depth map on our drone image. We can combine that with color as well to look at the canopy size. We've got a workflow where we identify the rows and then we take I think these are 30 centimetre distance data down the row and make an estimate of the fractional ground cover, which we can then trace down a row and see how it varies. And plot that over a field. So this is just the Google Earth image. This isn't a drone image or a good image. This is just from Google Earth. And if we plot over the fractional ground cover from our single drone run there, we can see, this is quite variable in terms of the soil here. And this image was taken 2-3 years previously, but we can see where we've got the high density of canopy versus the low. So again, that can be mapped out and potentially provided to the grower. So finally, in terms of the canopy data, if we take this bottom GoPro here and that looks up through the canopy against the sky, we can take each of those frames, we can colour segment them into leaves and sky and so on. We cut out the large areas of sky and then we identify all of the gaps in the canopy and apply a gap fraction analysis to that. Just to make our life easier with ground-based measures, we've also put a post-detection algorithm in there. And so then what we get is, this is the GoPro video looking up. This is the gap fraction detection. And then the post-detection gives us data for a panel. So we have continuous gap fraction or LAI data and then a per panel average. And obviously we could do that per meter or whatever. This is just to demonstrate. So that kind of covers all the work I was going to talk today about in detail. We're also, as I sort of suggested at the beginning, doing some work with the Hyperspec on canopy composition. So far this has basically been in the lab, but we're working with Fraunhofer over at Magdeburg in Germany, and so they have a hyperspectral aerial platform which gets mounted onto a fixed wing aircraft and flown over some of our sites. And so they also use their own machine learning algorithms to link with ground truth data in terms of the canopy composition. We've been doing some lab work on early detection of powdery mildew in leaves, and the problems it's looked promising at times, but the problems we're having with this is to have a good ground truth system. So If we use isolated leaves, for instance, it's difficult because the leaves are senescing at the same time that the disease is progressing. So we really done some initial testing on glasshouse plants, but that's as far as we are with that. And we've also done some basic lab testing of the fruit condition. So we got really good calibration, for instance, with shrivel in Shiraz, like on whole bunch data. But again, that's not been trialled in the field yet. So, as you might imagine, I'm just the person up here standing. Standing up here giving this talk. So Mark Thomas and I have been running these projects until Mark Thomas retired. On the ground, we also have Dom Mackenzie, who looks after a lot of the ground truth data work, plus a whole pile of other people who've helped out at times. We have Najid and Adam, who are doing a lot of our hardware development and some of our basic sort of coding work and software interfaces. They are looked after by Stephen Gensimer, who is in the manufacturing business unit of CSIRO and has moved down to Adelaide to assist working on this. So he's an optical physicist by background, but also very good with the hardware work. The LiDAR work is with Payman and Tom, and again, David Haddon helps with some of the software development. The machine learning and image analysis work is done by colleagues in Sydney. Rizwan is a postdoc who's been doing a lot of the machine learning work, although we had someone else assisting with that before, and Dadong and Chan Ming look after him. And then the radar work has been done by Chad and John O up in Brisbane, and their technical assistants, Alex Pitt. So there's a lot of people involved. And finally, we've got a job going. So we've got a junior experimental scientist position. So we're after someone who's got a background in I guess management and biology of perennial horticulture, but some experience in applying digital tools. So the level we're after is someone with a bit of postdoc experience who's ready to sort of move to the next level. So if you or anyone you know is interested in that, please go here or just search for the job at the CSIRO job site. We have good weather in Adelaide, so it's a good place to be. And that's it. Thank you.
Thank you, Everard, for this great seminar. Any question you have? I have one. You mentioned that regarding yield estimation, you know that the 60% of the variation is due to the number of fluorescence or bunches because you don't thin.
Yeah.
But how do you take into account the remaining 40% of variability within clusters?
Yeah, so with the radar work, that's basically will give us, if we get that to a quantitative position, that's going to be based on mass. So that will already give us that data and the spatial information. With the image analysis, as you suggest, we basically have to take the bunch data and find a way of looking at the bunch volume or bunch mass from that. So I mentioned that we're moving to having classifications for each of our hand labeling on the images. So one of the things we're looking at there is to take the bunches we've classified as completely visible and then potentially do some size and shape measurements on there. So I mean, like with other people, we've done work with image segmentation of bunches to count berries and find shapes and sizes and so on of bunches. So we can potentially apply some of that to the ones that we've classified as a whole bunch, get an estimate for average bunch size, and then multiply it up by the number we've got, where we've only seen part of the bunch. At the moment, this is where we've got to work out what works. And then the other thing is we can take the bounding box already. So that box that is drawn around the bunches, when the detector algorithm detects that, we can get the maximum dimensions of that box. And we've also done some work with more detailed shapes around, but it's just too long for the hand labeling. If we have those maximum box sizes and then we add depth information, whether it's through stereo or photogrammetry or something else, we can then take the bounding box, scale it according to how far it is, and again get an estimate of the bunch size. How accurate that will be, obviously we have to wait and see.
Would you have higher expectations with inflorescence or with whole bunches?
I mean, it's a lot harder to see all the bunches, so there's certainly more variability, and there's worse vineyards to be imaging in than the one I showed. Yeah, so I think the inflorescence is simpler because we can see them nearly all. So that's a matter of getting the timing right. So we've got maybe a 1 to 2 week window within a vineyard block where we can get see the majority of them. So I think that's simpler. In terms of the final yield estimate, our primary aim is to get per block estimates. So if we can get a good per block estimate, even if the spatial variation is not as accurate, that's what industry is interested in at the moment. Hopefully we can get good spatial estimates too. But so, I mean, I'm happy to talk to anyone who has any specific interests in areas another time.
Thank you again for having this nice presentation, and if anyone is interested in talking about specific issues o que alguien quiere hablar con él en algún aspecto concreto o algo, pues bueno, estaremos aquí un ratito pues, si alguien quiere acercarse o está interesado en la posición. Así que bueno, pues thank you very much, muchas gracias.
Thank you.